All right, ladies and gentlemen, boys and girls. Hopefully you guys got your notebooks out and you got a pack of colors, colored pencils or markers, because today we're going to do some pretty pictures as we start to talk about. We're going to start talking today about the structure of the cell membrane. We're going to dive deeper into some of the structures in the cell. But before we even do that, which I have on the board over there, before we do that though, I need to do a quick recap on, so hopefully something you guys brushed upon in physical science, although, if you remember physical science. All right, and this idea of what a solution is. All right, not the answer to a math problem. All right, a solution, right, is a mixture. It's a mixture. It's a homogeneous mixture. Basically means it's combining two things. All right, most common examples that you guys are probably familiar with are salt water and sugar water, or Kool-Aid, or lemonade. They're mixtures that, there's something in something else that's kind of being dissolved, right? And so in any solution, there's always two parts. And it's always hard because they all sound the same. In a solution is made up of a solute and a solvent. Not easy because they all sound like SOL, 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 all right? Solute and solvent. So there's basically two things of it. In a solution is when is a combination of two things, that one's dissolving another. All right. So a solute is what's being dissolved. And then the solvent is what's doing the dissolving. All right. So the classic example here is I have salt water. All right, so I drew my blue water, even though water's clear, but it represents blue. And then I have my little salt particles in there as well. All right, in this situation here, this is the solution. That solution is made up of two parts of the salt water. Is salt, which is the solute, and water is the solvent. All right, I'm going to tell you right now, water 99% of the time is the solvent. In fact, it is known as the universal solvent. In most solutions, water is that solvent. And especially when we talk about biology, because water is such an essential part of life. And part of it, the reason why, is because it is the solvent in the solutions that we find in cells, out of cells, all around life. All right. So real quick, recap, solutions made up of multiple things. Stuff being dissolved, typically in water, right? So the example, I always like to think of salt water. Solute is the salt. Water is the solvent, right? Because in your cells, if we're going to talk about the cell membrane, in your cells are all kinds of solutions. You have a solution on the outside of your cells and a solution inside your cells, all right? So let's go ahead and hop over here. Pause it if you need to take a second. Okay, we're ready to move over here. And now we can start talking about the cell membrane. Get myself nice and set up here. All right, now remember the role of the cell membrane, its job is to allow certain things in and out of the cell. And if we have a solution, right, on the outside of the cell and on the inside of the cell, so there's a solution out here. This is my cell. I have a solution on the outside of the cell, and I have a solution on the inside of the cell. I'm going to want to have solutes and solvents go in and out of my cell. All right, so I'm going to need to have things go into the cell and things come out of the cell. And it has to go through the plasma membrane to do that, or the cell membrane. And so we're going to talk about the very basic structure of that cell membrane. And then using that basic structure, we can talk more about its functions. Remember, structure determines function. So today we're just going to talk about the structure, and then the next couple times we'll talk about the actual function of that cell membrane. General function being what lets in and out things in and out of the cell. So if I took this little part right here, I have all my organelles in here. I got my cell membrane along the outside. This is obviously an animal cell because I do not have a cell wall. All right, and I just zoom in just on that part, and I'm going to blow it up so I can look at it here. This is kind of what it's going to look like, all right? What we have here is let me talk a little bit about the structure here. We have what looks like these two layers of these these circly things. and then we have these big things sticking into it, and actually we'll have a couple of these almost like this twisty, curly things kind of sticking up through them every once in a while. All right, let's talk a little bit about the structure of what we see here. Let's start with the most important part. It's the little black parts here. All right, these are what we call phospholipids. All right, so I'm going to even take this one right here. I'm going to draw bigger over here. All right. All right, they look like that. All right, there's a bunch of them on there. All right, we have, excuse me, we have two parts to this. We have the little head, and then we have these little tails. All right, this is what we call the phosphate head. And then we have below it, we have lipid tails. Now remember, lipids are a type of macromolecule. They're like fats and oils. All right. All right. We have lipids, tails, and because of that, we call each one of these things a phospholipid. All right. Now, if you look at the way I have my phospholipids set up on my little diagram here, you'll notice something. First of all, a couple things you should notice. There's two layers. All right. More importantly, they're not just stacked on top of each other. Look how they're oriented. The tails are facing each other. All right. And that's because we have, again, just as my picture here, here's the outside of my cell. Here's the inside of my cell. All right. There's a solution out here. And there's a solution out here, or in there. That solution is made up of a solute, which those solutes could be lots of things. It could be glucose molecules, it could be proteins, it could be amino acids, it could be free-floating nucleotides, it could be all kinds of stuff. It could be other phospholipids, all right, just kind of floating, free-floating, all right? These tails are lipids, like a fat and an oil. All right, those solutions are made up of a solute and a solvent, which is water. These tails are what we call hydrophobic. Right? Hydro meaning water, phobic meaning fear. Right? These are hydrophobic. These are hydrophobic, which means they don't like water. All right? And these ones here are hydrophilic. Philic meaning like. Right? They like water. All right? Which means, notice that all the phosphate heads are facing towards the solution. Those solutions have water in them. The tails are facing, why are the tails facing inward? Right? The tails are facing inward because the tails are avoiding the water in the solution. Right? And why are the heads facing out? Because they chemically want to be attracted to the water on the outside. All right? This prevents water from just going in and out of the cell. Water can't just travel into the cell because once it hits those, it's attracting those phospho heads, but it's repelling those lipids. Water can't get in because those tails are afraid of water. They won't let water just go in. All right? Since they won't let things just go in a lot of times, all right? These things that, like water, they have to have other things to allow certain things to go in and out of the cell because that's the whole idea is let things in and out of the cell. So what they have is these proteins, right? That's what we have here. And there's all different kinds of proteins that are on here, All right. These two here I drew as these are what we call channel proteins. Water can't get through that membrane because those phospholipids, or because the lipids. All right. So what they have to do is they need a tunnel, and so that water might come through this tunnel here. We'll talk a little bit more. That's called osmosis. All right. So we have these different types of proteins embedded throughout them to help us understand a little bit more about the to let things in and out of the cell. And we're going to talk about that process going on. But it's because those structures, those phospholipids. And then we will typically embedded throughout this whole thing. We have these little things. We have cholesterol. All right. That serve multiple purpose, right? Some proteins are used for transport. Some proteins are used for signaling. Cholesterol's job gives it this fluid motion. All right, cholesterol is a type of fat. It's a type of lipid, right? And it gives it that fluid bubble-like motion. All right. So this here, all right, we have a very fluid-like barrier. All right, that allows certain things in, but not everything. All right, it allows some things in, especially through the protein channels, but not everything. So it's not completely permeable, but it's also it's not a wall. This is what we call semi-permeable. It allows some things in, but not all things. Or what we call selective permeability. It allows some things in and some things are not allowed in. And we'll talk more process about how that actually happens as well. All right. Because of this, the cell membrane is sometimes also known as, based on the structure that we have here, sometimes also known as the phospholipid bilayer. It is 2 layers of phospholipids. And it's based on those lipids are oriented because of the way the water's set up in the solution. Okay, so quick recap to end this here. All right, we have that cell membrane that determines what goes in and out. All right, when we look at that membrane, we see this very fluid-like material. All right, that's mostly made up of these things called phospholipids that have a hydrophilic tail or head and a hydrophobic lipid tails. Because of that, hydrophilic, it only allows certain molecules in and out. That makes it what we call semi-permeable. Right? Now, embedded throughout that entire phospholipid bilayer, all right, is proteins and cholesterol. All right, those proteins can either act as a channel. All right, they either can do it for transport, or the most other predominant one is for signaling, so they can talk to other cells, especially if they are multicellular. All right, that is our setup there. Woo! We're going to be drawing a lot of pictures. I hope you got lots of colors on your page too, because we're going to keep them going. All right. Questions to think about at the end. All right. What is a phospholipid? Why is it called the phospholipid bilayer? Why do the tails face on the inside? What's the purpose of the proteins? All right. So things to think about a little bit there as we kind of keep going. What does it mean by semi-permeability? What does that mean? If you don't know the answer to any of those questions, or you weren't trying to answer them just now, go back 10 seconds and listen to those questions again. You should be able to answer those questions. If you don't, go back even further and figure that out. All right. Otherwise, we're going to be doing a lot of phospholipid bilayers. We're going to talk about what goes in and out of the cell, but that's enough for right now. So good luck, and may the science be with you.